lymph is not the same thing as blood. All right, but lymph resembles a component of blood, doesn't it? Which component?
Plasma.
Plasma. So it resembles plasma, but it's not exactly plasma either. Because most of the proteins remain in the bloodstream. But some of the smaller proteins can get out of capillaries as that blood flows from arteries into capillaries, into veins, and back toward the heart. So it has a lower concentration of proteins than plasma has, but you know, it's very much like plasma anyway, because before it was lymph, it was plasma. You understand that part? Because the heart is pumping blood under pressure, and that pressure is forcing, when it gets to the capillary level, it's forcing some of that plasma and maybe a few of the small proteins outside the capillaries. And so it pushes them between those intercellular clefts. Now, most of that fluid that's pushed out by that pressure is gathered back in by the osmotic pressure created by those plasma proteins. But not all of it makes it back. So what doesn't make it back is picked up by those lymphatic capillaries, because you saw earlier that those lymphatic capillaries are all intertwined with the blood capillaries. So that's why that's so. So normally, lymph is clear in color, kind of watery-like. After a person has eaten a meal with a high fat content, and that lymph is gathered from the intestinal tract, it can have sort of a milky look, like very dilute milk. Now, on its way back toward the heart, this lymph is filtered by a number of lymph nodes, because that lymph is flowing through these filters, which are the lymph nodes. And so there can be a number of those lymph nodes in the route from some point in the body back toward close to the heart. So it's constantly being filtered by those lymph nodes. And by the way, the only lymphoid organs that filter lymph are the lymph nodes. The spleen does not filter lymph. Tonsils do not filter lymph. The thymus does not filter lymph. Only the lymph nodes. All right, so now, blood has a pump that moves it through the blood circuit, right? The heart's the pump. Is there a lymph pump? No, no such thing. So how then does lymph find its way back in the direction of the heart? Well, it actually moves a lot like blood moves through veins. So the milking action of skeletal muscles helps blood move back through veins to the heart. And since these capillaries lie along blood vessels and deep to muscle, the milking action of skeletal muscles when we move our skeletal muscles helps to milk it back in the direction of the heart. These lymphatic vessels have valves in them, just like some of our veins do. So veins in our lower extremities have valves in them, so that milking action of the muscles as it milks it toward the heart, those valves keep that blood from sinking back by gravity toward the feet. Well, we have valves in our lymphatic vessels too that do the same thing. Remember now, these lymphatic capillaries are blind-ended, so they're not hooked to a pump. All right, a lot of these lymphatic vessels lie along big arteries, along the aorta and its major branches. and the slight pulsing action of the aorta and its major vessels will help to move lymph along too. Something called the respiratory pump helps lymph move along, just the way it helps blood move along. So blood from our lower limbs and our abdominal cavity is moved toward the heart by the breathing action, the moving of our diaphragm up and down. So when we take a breath in, we reduce the pressure in our thoracic cavity, and that slightly reduced pressure facilitates the movement of blood toward the heart. Then when we breathe out and that diaphragm goes up, that increased pressure that's already drawn a little bit of blood and lymph toward the heart then pushes it further until we take another breath. So this is why this is in quotes. It's acting like a pump, the in and out movement of our diaphragm as we breathe. Then in some of the larger lymphatic vessels, we do have some smooth muscle that helps the lymph move along too, by those muscle contractions. All right, so over a period of 24 hours, how much plasma is pushed out of capillaries that doesn't make it back into those capillaries? If you total it all up over a 24-hour period, we'd have close to 3 liters of fluid. That's a significant amount, isn't it? When you think that in an adult male, the circulating blood volume is 5 to 6 liters, well, that's a significant amount, isn't it? So the recovery of that lymph to the blood circulation, dumping it back into blood circulation, helps maintain our cardiovascular status. It helps us keep our blood pressure up and our cardiac output up. Because if you lose a unit or 2 units of blood, is that going to tend to affect your blood pressure and your heart output? Yeah, it is. So we're recovering this fluid here. All right, now lymphedema. Let's talk about edema first. Edema is just a collection of fluid in places where it shouldn't be. So somebody with congestive heart failure has a weak heart. They're not able to pump all the blood effectively that is introduced to it. Creates a back pressure. That pressure builds up in the lungs. They tend to get short of breath because they're pushing some fluid out into the air sacs. And then sometimes they'll get swelling in their abdominal cavity, ascites. Sometimes they'll get swelling of the feet and ankles because you've got backup. That's edema. That's edema of congestive heart failure. Okay, so edema is the broad term. Lymphedema is one type of edema. So lymphedema is the collection of lymph in places where it shouldn't be collecting. So it results from obstruction to the flow of lymph. Anything that will impede the flow of lymph will create a buildup of lymph proximal or behind the obstruction. So years ago, back in the 50s and 60s, even in the early 70s, the treatment for cancer of the breast in women was commonly to remove the breast on the affected side and the pectoralis major muscle and the axillary lymph nodes under the arm on the affected side. Well, a number of these patients subsequently would develop swelling in the upper limb on the operated side. From what? Lymphedema. Because the lymph that normally used to flow through these lymph nodes on back toward the heart now is blocked from doing that because those lymph nodes have been removed and there's been some scarring in there and it's sealed down a lot of those lymphatic vessels. They tend to get swelling in the arm or upper limb. You know, you can you can overcome this by application of a specially fitted pressure gradient sleeve that keeps the, the lymph out. Another example might be from, say, a parasitic infestation. Let's say a person has a parasitic infestation, say, in the groin nodes, the inguinal nodes down here in the groin. It's not common in this country, but in some countries you can have this. One such type of parasitic infestation is called filariasis, where these worms get into the lymph nodes and clog up the lymph nodes. All right, that obstructs the flow of lymph out of the limb on toward the heart. And these folks tend to get massive swelling in the limb on the affected side. And this is a chronic thing, and in the extreme, it can make the limb really very swollen, especially in the leg and the foot. The skin becomes thick and brawny and rough looking and discolored, and in the extreme it can even look like an elephant's leg, thus the name elephantiasis. You may have seen pictures of this. Okay. All right, lymphangitis. Lymphangitis. Okay, remember we said anytime you see itis on the end of a word, it means inflammation of. All right, it doesn't mean infection of necessarily, but inflammation of. So the A-N-G here stands for vessel. So lymph vessels that are inflamed are called lymphangitis. Now you may have seen a case of this, may have even happened to you. Let's say someone has an acute bacterial infection in the hand. And the lymph drainage from the hand is going to contain some pathogens and other cells of inflammation. And of course, this is infection. Infection is inflammation plus the presence of a pathogen. All right, so you're going to be draining pathogens away from the hand up through the lymph vessels this way. And sometimes that will cause red streaking in the limb. You can see the red streak. All right, now it's called lymphangitis, but what causes the red is the dilation of very tiny blood vessels in the wall of the accompanying blood vessels. So it's the blood vessels that are causing the red streak, but the red streak in the blood vessels is caused by the pathogens and the infection in the associated lymph vessel. In other words, it's causing the tiny vessels in the wall of the blood vessel to dilate, and it shows up as a red streaking that you can see through the skin. All right, we talked about the lymphatic vessels being important for maintaining stable circulating blood volume. All right, so let's go on to the cells of the lymphatic system. And these are the cells of the lymphatic system. So first on the list is lymphocytes. That's pretty logical, isn't it? Lymphocytes in the lymphatic system. All right, and then we got plasma cells and macrophages. Where do macrophages come from? Monocytes. Then we got some strange cells called dendritic cells, and then we got the cells that form the stroma, the reticular cells. All right, lymphocytes. By now, you should know for sure what a lymphocyte looks like. Now, this would be a small lymphocyte. Some are larger than this, but most lymphocytes are about the size of a red blood cell. Large nucleus, little thin rim of cytoplasm. Now, where do these lymphocytes come from? Bone marrow. What was the the common stem cell of origin, hemocytoblast. All white cells come from that, and red cells too. In normal blood, these lymphocytes make up about 25 to 40% of the circulating white blood cells. Most of them, however, are in tissues other than blood. In total number, we have about 10 trillion. So out of our 75 trillion or so total cells in the body, about 10 trillion of them are lymphocytes. They have a very long lifespan, years sometimes. But a lot of them typically last for decades, as opposed to, say, neutrophils that have a lifespan just a few hours. And when they're actively engaging pathogens, it's minutes, seconds. All right, this fact right here has an awful lot to do with the fact that you can have immunity against a foreign antigen that lasts a long, long time. So you have a vaccination for something, immunization that can last a long, long time before you have to have a booster. Now there are three classes of lymphocytes: T cells, B cells, and NK cells. The T stands for thymus. We'll find out more about that later. The B stands for bone marrow. We'll find out more about that later. The NK stands for natural killer. We'll find out more about that later. Now, when we're looking at our slides in the lab, and we see these lymphocytes, is there anything about the lymphocytes we can see on those slides in the lab that will tell us whether they're one of these three kinds?
That level of magnification.
No, absolutely not. On a light microscope, all you can tell is it's a lymphocyte. So they're very refined lab procedures, techniques, whereby you can sort these out, and it's commonly done. Commonly done. So the lymphocytes are the main cells of the immune system. Now, in lymph nodes, there are lots and lots of lymphocytes, but that's not the only place that you'll find them. You'll find them in other tissues besides lymph nodes, and of course you'll find them circulating in the bloodstream too. Of course, blood is a tissue. All right, next on our list is plasma cells. We'll find out more about plasma cells later, but plasma cells come from B lymphocytes. So we start with the B lymphocyte. And when we encounter a foreign antigen that group of B lymphocytes is specific for, we will form plasma cells off of that B lymphocyte population. And those plasma cells are the cells that actually make the antibodies. Antibodies come from B lymphocytes, B cells. But the cells that actually produce the antibodies are an intermediary cell. So you got B lymphocyte, plasma cell, antibodies. All right, and then we have macrophages. This is an electron photomicrograph of a macrophage actively engaging bacteria, pathogens. So this is extremely highly magnified, but you see the cytoplasmic extensions whereby this macrophage here is engulfing the foreign antigen and it's going to bring it into itself, into its cytoplasm, and break it down. So we said before that they come from monocytes. They're highly phagocytic. They're very prevalent throughout the body. There are lots and lots of them in our various lymphoid organs and even in the liver, but a lot of them are roaming outside those tissues as well. These macrophages have a major role in the immune response and in the inflammatory response in general. So whenever there's inflammation, you'll have monocytes, which have been converted into macrophages. All right, and then this strange creature here is a dendritic cell. This is also an electron photomicrograph, highly magnified. Now, what do these dendritic cells do? Well, their main role is in what we call antigen presentation. In other words, they present antigens to some of our immune cells, particularly to the T cells. So they are in a class called antigen-presenting cells. So dendritic cells are antigen-presenting cells, macrophages are, and also the B lymphocytes are, as we'll explain later. Where do we find them? We find them in lots of various connective tissues. We even find them in the epidermis of the skin. So, and when you looked at skin, and if you happen to see some Langerhans cells, does that name ring a bell? Langerhans cells, those are dendritic cells. So what these cells do is they pick up antigens and take them back to where there are large aggregates of lymphocytes, like in lymph nodes, for example, so that they can be presented to the immune cells. Then we have these cells here called reticular cells. These cells create the fiber network, which is the skeletal framework of lymphoid tissue and lymphoid organs. So reticular cells create this mesh network here, which is the stroma. Now, hanging out on this stroma, you have lots of the cells of the immune system. So you have some lymphocytes and some macrophages hanging out here. Now, if this were a lymph node, for example, this would be the skeletal framework of it. These spaces in here might contain lymph. Lymph will be percolating down through these fibers. Now, when we look at our lymph nodes and spleen and so forth in the lab, when we get to that in lab, are you going to be able to define the reticular fibers when we look at our slides? No. Because in order to see reticular fibers requires a special stain. They make special stains that bring out the reticular fibers. If you put a reticular fiber stain on, say, a lymph node, and put that under the microscope, you're going to see lots and lots of crisscrossing black fibers there, which will be these fibers that are shown in blue right here. Reticular fibers typically stain black on the common stains. All right. Now, going on, so we talked about lymph itself, the cells of lymph. Now, let's talk about lymphoid tissue. Now, in a minute, we're going to get to lymphoid organs. Lymph nodes, spleen, thymus, tonsils. But it is true those organs are lymphoid tissue. But those organs have a capsule. In other words, it's all contained within a capsule. But there is lymphoid tissue scattered about the body in many, many places as little aggregates, little collections of lymphoid tissue. Here is just one example. And you'll see this when we get to the digestive system. This is a cross-section of small intestine, part of it. You just see the outer part of it. The lumen of the intestine is over here. But you see these little black speckled nests in here? Those are lymphoid nodules or lymphoid follicles. What are these little black specks? They're probably the nuclei of lymphocytes. So these are all little patches in here, little collections of lymphocytes. They have a particular purpose by being in here. So lymphoid tissue is typically non-encapsulated. When you get to the lymphoid organs, like lymph nodes and spleen, there will be a capsule. Now, we have different types. As I told you, this lymphoid tissue is diffusely arranged in the body, and it's in virtually every organ. These lymphoid nodules or follicles are just aggregates of lymphoid tissue. What is lymphoid tissue? It's reticular fibers containing lymphoid cells, lymphocytes, macrophages, dendritic cells, so forth. Now, so we find it in many, many different places. This acronym right here stands for mucosa-associated lymphoid tissue. So in our major organ tracts, like our respiratory tract, our digestive tract, our urinary tract, our reproductive tract, deep to the epithelial lining, as part of the mucosa, you will typically find many, many, many little collections of lymphoid tissue. So this is where lymphocytes hang out, and because they're hanging out there, they're able to monitor the tissues, the fluid that's passing by them, for foreign antigens. Okay, that's what they're there for, to act against foreign things that can harm us. All right, and then we have our organs. So the lymphoid organs are just larger aggregates confined and inside a capsule. Okay, the current scanning plans. Would you like to cancel this process? I'm going to click yes. All right, so lymph nodes, which are not shown in this diagram, spleen, thymus, tonsils. Those are the lymphoid organs. All right, now of course this just abbreviated diagram here doesn't show you nearly all the lymph nodes by any means. It's just showing you in general. All right, first the lymph nodes. So we know that they're numerous. They're usually embedded in connective tissue, and that connective tissue is most commonly adipose tissue. It's very common to find lymph nodes embedded in fat, wherever you might find them. Now, as we said, these lymph nodes are filtering devices. They're filtering the lymph as it's passing through them. So they're going to pick up foreign antigens, toxins, various debris, and so forth. And because we've got many, many lymphocytes residing in those lymph nodes or passing through them, those lymphocytes are able to recognize foreign antigens as they pass through. And as a result of that, our immune response is activated.
Hey, Professor, I have a question. So, okay, I understand that they filter the lymph nodes, filter lymph, but do most of these, what do you call it, foreign material, does it come, is it in the bloodstream, or is it coming from other parts of the body when it goes into the, lymph?
It can be in the bloodstream because these lymph nodes have a blood supply. So lymph, passes through the lymph nodes, and we can pick up foreign antigens that way. But if there is a foreign antigen in the bloodstream, it's going to find its way in the lymph nodes too, because lymph nodes have a blood supply. In fact, a lot of the lymphocytes get into lymph nodes through the bloodstream as opposed to coming through the lymphatic system. So you can pick up a foreign antigen in your bloodstream when you cut yourself, or you have, something, say you have an injection injury. You've got a hydraulic pump that pumps something foreign into your hand. Well, that stuff is going to tend to be picked up in the lymphatic system. Does that make sense?
Yes. Thank you.
All right, so what about the anatomy of a lymph node? Well, lymph nodes in size tend to be somewhere between 1/2 to 3/4 of an inch. Some are smaller, some are bigger. In the living state, they tend to have a grayish color, and they're shaped like a kidney, or a kidney bean, typically. As I said, they're embedded in fat commonly. And they have a blood supply. All right, so that's grossly what they look like. I'm going to get to the microscopic anatomy in a moment. But talking about these lymph nodes, we know that they're scattered through the body diffusely, but there tend to be major collections of them in certain parts of the body, especially where the limbs join the trunk, where the neck joins the trunk, and so forth. So in these regions here, we give them names. So in our neck here, we call these cervical nodes. Under the arm, we call them axillary nodes. Inguinal in the groin, we call them groin nodes or inguinal nodes. We got a lot of lymph nodes in our mediastinum right here, centrally. Some of them get to be quite large. Those are bronchomediastinal nodes. We got a lot of nodes along blood vessels, especially major arteries. Those are called perivascular nodes. When we get to talking about the digestive tract, we're going to be talking about the mesentery. The mesentery is what the digestive tract is held on, where it gets its blood supply and so forth. We'll talk about that. But there are lots of lymph nodes associated with the digestive tract in that mesentery. So that term may be foreign to you right now, but I'll guarantee you we're going to have you understand what a mesentery and mesenteric nodes are later on. All right, and then other nodes scattered, they're not shown. All right, so these lymph nodes, they filter the lymph, and they're recognizing and picking up foreign antigens. They're also recognizing our self-antigens too. All right, so here is a diagram of a lymph node. So it has a capsule here. And you see these vessels here going into the node? Those are called afferent lymphatic vessels. Afferent means going toward or to, doesn't it? So remember I told you that this lymph is passing through those lymphatic vessels and it's filtered by the nodes. And that lymph is going to enter through these afferent vessels. It's going to percolate down through the node, and it's going to come out these vessels heading on upstream to the next node. These are efferent lymphatic vessels. Efferent means going away from, doesn't it? All right, now, what is not shown on this diagram, and I don't know why they didn't show it, is the blood supply. Because every lymph node does, in fact, have a small artery feeding it and a small vein draining it. If you ever try to pluck a lymph node out of your way when you're dissecting without controlling its blood supply, it is going to bleed every time because they all have a blood supply. All right, now, inside here, what have we got? Well, the inside of a lymph node is divided into two main regions. The outer part is called the cortex. The inner part is called the medulla. Now, lymph nodes are not the only organs that have a cortex and a medulla. Okay? Kidneys have a cortex and a medulla. The adrenal gland has a cortex and a medulla. The thymus has a cortex and a medulla. It's just the cortex is typically the outer part of an organ. The medulla is a central part of an organ. So here, right here, from here out, would be cortex. Down here is medulla. Now this capsule here is a loosely arranged connective tissue, but it sends little strands down into the cortex like this that divides the cortex off into these regions. These strands are called trabeculae, but it divides the cortex off into these lymphoid follicles. So each one of these is a lymphoid follicle. Typically what you'll see in these lymphoid follicles is a more central zone of cells that take a different stain. Usually it's lighter, sometimes darker. These are called germinal centers. What happens in these germinal centers?
The actual filtering.
Production of more lymphocytes, cloning of lymphocytes. Now, where did we say lymphocytes come from originally?
Bone marrow.
Bone marrow.
They mature.
But once they're formed, they can reproduce themselves within lymph nodes. All right, so you can have cloning here of a certain population of lymphocytes that are specific for a certain antigen. Say you've got a foreign antigen coming through here. It's picked up by the neighborhood of lymphocytes that it's specific for, and these lymphocytes then clone themselves and make more of their own kind in order to respond to this foreign antigen. All right, so you've got lots of lymphocytes here. Mostly out here you got B lymphocytes, a little deeper you got T's, but down in here in the medulla, you don't have lymphoid follicles anymore, but what you have is little strands of cords called medullary cords surrounded by open spaces called medullary sinuses. So the lymph is going to filter through, and typically down in this zone there are lots of macrophages and also antibodies right in here.
I have a question. On the lymphocytes, I know they're produced in the bone marrow, but we also said that they mature in the thymus?
The T cells mature in the thymus.
Okay. Okay, just the T cell. The
Just
thymus.
the T cells.
And that's different than...
That's different from
Because
this.
these are the
Now,
Ps.
now there can be T cells in here,
Okay.
but those T cells that are formed in the bone marrow, when they come out of the bone marrow, newly formed, they pass through the thymus,
Okay.
where they're programmed
Okay.
and selected out. In
Okay.
other words, the ones that don't meet the necessary requirements are bumped off, so that hopefully we just retain the ones that will be able to recognize foreign antigens and attack them while recognizing our self-antigens and leaving them alone.
Gotcha.
All right. That's only the T cells.
T cells.
The B cells mature in the bone marrow,
Okay.
so it's a different process for them. So thymus is just for T cells.
Okay.
I have a question. Back when they're cloning themselves, can any mutations happen during that process?
I would think it'd be possible, but not
Not like
likely. Not likely. But mutations can happen, but probably when lymphocytes are cloning against a specific antigen, probably not. I think it'd be very rare if it did happen. I don't know that it can happen. All right, so this is just another view. This shows you the thin capsule here, little connective tissue extensions called trabeculae. So this is a lymphoid follicle, and this darker staining area here is a germinal center. Now this shows you the medullary cords and sinuses in the medulla down here. This is just a piece of it. See how this looks different from this? See these solid green things? Those are the medullary cords, that's solid tissue. This lighter green is lymph. That's just lymph. Now the lymph is passing through here too, but there's so many cells you don't appreciate it. But down here you got a little more space. All right, so going on to the spleen, another lymphoid organ. We're going to talk about the anatomy in a moment, but let's look at the functions of the spleen right here. The spleen is a major blood filter, is it not? Because the spleen is the primary organ that takes out of circulation aged and or damaged red blood cells. It's not the only organ that can do that. The liver can do it too, but the spleen is the major organ. Now we're going to find that the reason the spleen is classified as a lymphoid organ is because it has lymphoid tissue in it. Now, its main function is filtering of the blood, but it does have some lymphoid tissue. The spleen also stores platelets. So of all of our platelets, about 2/3 are circulating in the blood, but about 1/3 are stored in the spleen. And if you'll recall that the spleen had a role in blood cell formation in the fetus between the 2nd and 5th months. It and the liver were the main sites of blood cell production. But after that, the bone marrow took over. Just a word about splenic trauma. It used to be, in times past, that if you had a ruptured spleen from an injury, out the spleen came. Every time. Ruptured spleen, out it goes. Or out it comes. Now, there's a more conservative approach to splenic trauma, and just because you have a ruptured spleen doesn't necessarily mean it has to come out. Since it's a useful organ, it would be nice if we could keep it, if we could, right? So nowadays, if someone has a ruptured spleen, they come in the emergency room, they're evaluated, and say they're hemodynamically stable, but they've lost some blood, but their vital signs are still stable. They've resuscitated them with some fluid and so forth, maybe even a transfusion. But they become stable and are remaining stable. And put them in intensive care and they monitor them very closely. Monitor the vital signs, urine output, and do periodic hematocrits, checking the blood count. If the blood count stays stable and over a period of time shows a tendency to rise back, then they leave the spleen in. The tear heals, and these people leave with their spleen. So, this happened a few years back to a backup quarterback for the Tennessee Titans, National Football League, Billy Volek. So as far as I know, Billy still has his spleen today. All right, here's a picture of the spleen, and that's kind of what they look like. They have a dark grayish color. Where's the spleen, by the way? Up here, under ribs 9, 10, and 11, right up here. All right. Now, that is a big artery, isn't it? That's a splenic artery. That thing is carrying a lot of blood. All right, so blood goes in arterial-wise here, like this. It's filtered out and passes into the venous side and back out. There's an interesting arrangement in here. These are very special capillaries inside the spleen, unlike capillaries throughout the rest of the body. These capillaries, their walls are formed largely by macrophages. And so the blood has to pass through a phalanx of macrophages. And the normal blood cells are allowed to come through. But the abnormal cells, the aged and or damaged red cells, are phagocytized by the macrophages and pulled out of circulation. So the normal red cells pass through into the venous side and back out the spleen. The aged and damaged red cells are taken out of circulation there. Now, here is a photomicrograph of spleen right here. Now, all of this that you see in here that looks sort of homogeneous, that's the red pulp of spleen, histologically. The red pulp is the blood filtering part of the spleen. But you also notice scattered through here, you have some islands of darker staining cells. You see these? This is known as white pulp. This is the immune part of the spleen. So, in the total architecture, histologic architecture of the spleen, about 80% of it is red pulp, about 20% is white pulp. And so you look at this picture here and you say, I don't know where you get white pulp out of this. This thing looks awful dark blue to me. How come it's called white pulp? Well, it's called white pulp because the cell populations here are white blood cells. These are lymphocytes. Now, the dark color here is that you're actually seeing the nuclei of many lymphocytes, and nuclei take a very dark stain when you crowd them all up like that. So these are islands of lymphoid tissue within spleen. So spleen in this role has the same role as, say, a lymph node does. So that's what white pulp is.
I have a question on the spleen. It's kind of a silly analogy, but when I was a kid, we used to butcher chickens, and you always had to be careful not to rupture the spleen when you were cleaning them out. And this is what I was told when I was a kid, because if you rupture the spleen, it would ruin the meat in the chicken. But when we're talking about it here, we're talking about it basically filtering bloods. I was just always, I believed that it was filled with toxins and poisons for that reason. No? I mean, it's just, I'm just being curious, I guess, at this point.
I guess that might be the reason.
Okay.
You know,
Maybe my dad was just crazy and just didn't want me to pop his spleen in the chicken. Who knows? Okay.
yeah.
It's true.
Well, I used to kill a lot of chickens too, but I don't remember the spleen being.
It's like a little bluish greenish colored ball. And I always remember being told it was like a marble because we'd want to play with it.
Yeah, it must be.
Yeah, it's true.
Must be, not to soil the meat or something. I don't know if it's an old wives' tale or whether,
Maybe, yeah.
There's probably some substance to it, but I just don't really know the answer to the question.
So when I'm looking at that, I'm like, that doesn't look like a... what I remember, but okay.
Here's a bigger view of what we were just looking at there. Actually, my brackets should be down around here. I think I played with this or something. We couldn't get it back. But this is white pulp. All right, so we went over anatomy and the functions. Now the thymus. Okay, so where's our thymus now? Right here, isn't it? Lower anterior neck, upper mediastinum. All right? So right here. All right. Now, it's classified as a lymphoid organ, but it is unlike lymph nodes and spleen for some reasons that I'll tell you. But its main function is T lymphocyte maturation. What goes on here in the thymus relative to T cells is a process known as the development of immunocompetence, meaning, as I indicated earlier, These T lymphocytes that come out of the bone marrow pass through the thymus. They're subjected to a couple of important tests. They are tested to see if they will recognize foreign antigens and have the capacity to attack foreign antigens, while at the same time being able to recognize self-antigens and leave those alone. Because we're going to find out in the immune response that our T lymphocytes have to identify both self and foreign antigens before they attack. All right, so this is what happens in the thymus. So any self-reacted T cells are removed by a process called apoptosis, programmed cell death. Now, in a perfect world, this would be exactly what happens. But as you may know, there is such a thing as autoimmunity, when a person's immune system will begin to attack parts of itself which should not be attacked. And one of the theories is that one of the causes of autoimmunity is that some of these T cells slipped through this initial testing period right here and are circulating free. All right, now the thymus has some things that are unique to it. The main thing we said is for the development of immunocompetence in T cells. All right, so thymus does not directly fight antigens. Lymph nodes do. Lymphoid tissue does. Spleen does. Tonsils do. Thymus does not. The thymus does not contain phagocytes. And its stroma is epithelial cells, not reticular cells. We saw that the stroma of other lymphoid organs is reticular cells, the framework. That's because thymus derives from a different germ layer. In other words, the thymus is an out-pocketing or out-pouching of our primitive digestive tube. So its stromal cells are epithelial cells, not reticular cells. And finally, and this is not extremely important except to help you identify thymus histologically, because in the medulla of the thymus, as you will see here, there are scattered collections of these bodies known as thymic corpuscles or Hassel's corpuscles or Hassel bodies. All they are little swirls of epithelial cells, in sort of a concentric ring like that. They don't have any special function except to help you identify what's the thymus. Because if you see this, it's got to be thymus. It can't be anything else. And so in lab you'll be looking at slides that show you this. All right, and finally we got our tonsils. So our tonsils are located up here, aren't they? Now, why would we have lymphoid tissue up here? To filter the air that we breathe and the food that we ingest, right? Got, sizable collections of lymphoid tissue right there. All right, now we've got names for these collections of tonsils. So when you look in the mouth, right here, you see there's tonsillar tissue here and here as you approach the oral pharynx back there. Those are the palatine tonsils. If you've had your tonsils removed, these are the ones that were removed. All right, up here, right here, in your nasopharynx, you've got a collection of lymphoid tissue as well. And you can see that they're also calling this palatine tonsil. Boo-boo. This is a pharyngeal tonsil, so it's mislabeled. And then there's some tonsillar tissue on the back of the tongue called the lingual tonsil right there. All right, and so you'll be looking at these, demonstrations of these in the lab. So it's non-encapsulated tissue, contains both T cells and B cells, and of course the function is to filter pathogens that pass, that we breathe in, or in food and drink that we ingest. So if they're helpful in that regard, why do we sometimes remove them?
Why don't they get inflamed, but
Chronic.
Yeah, just chronic recurrent bouts of tonsillitis with associated morbidity, having to take antibiotics. Sometimes it's just better to remove them. Well, by the way, let me go back here. This collection of tonsillar tissue right here is often called adenoids. So if you've had your tonsils and adenoids removed, if you had a TNA, you had both of these and this removed. This can be a problem in young kids sometimes because this can be quite sizable and it can give partial nasal airway obstruction. So these kids will be mouth breathing a lot, and it's alleviated by removing that. This is just a histological view of tonsil. All right, so that's the lymphatic system.